This is our penultimate PowerPoint of the semester. We're going to look at evolution from the beginning of Earth until the development of primates. And our final PowerPoint will provide a brief introduction into the concepts that drive the process of evolution. Now, however, we're going to consider evolution from the beginning of Earth until the development of cells, and from there, the development of multicellular organisms. The evolution of cells, this is essentially the take-home message, involves three separate steps. The first is we have to produce biologic molecules, and the hypothesized first biologic molecule that led to the evolution of cells was RNA. Following the development of biologic molecules, it was necessary to enclose those molecules in some sort of container. We are going to presume that that container was originally a membrane, however, we don't know. And then finally, in order to develop eukaryotic cells, remember eukaryotic cells are those like we have that contain a nucleus. It was necessary to develop organelles, and the first organelle thought to arise was mitochondria, and the development of mitochondria resulted from the phagocytosis of an oxygen-utilizing bacterium. We're going to talk about each of these steps now briefly. Let's start, however, by looking at an evolutionary timescale. So on this image, you can see that the Big Bang was hypothesized to have happened about 13, almost 14 billion years ago. Following the Big Bang, there were a lot of things that happened, but for us, one of those important steps was the development of our solar system. And among our solar system, obviously, is our planet Earth. The solar system and Earth was hypothesized to have arisen somewhere between 4 and 5 billion years ago. Life on Earth, however, occurred roughly 3.5 to 4 billion years ago. In between the development of Earth and the development of life on Earth, the environment within that time frame was comprised mostly of carbon dioxide, methane, ammonia, nitrogen, hydrogen, and a little bit of water vapor. It was hot. The first four of these chemical compounds, carbon dioxide, methane, and ammonia, are not really conducive to life as we know it, but this is what at least is hypothesized to have comprised the atmosphere during that time frame. How did life develop from that environment where life as we know it could not exist? Stanley Miller in the 1950s, so approximately 70 years ago, demonstrated the theory of chemical evolution. This theory shows that the chemicals that were on Earth, those carbon dioxide, methane, ammonia, etc., could have produced biologic molecules. And the way he demonstrated this was the contraption shown in this image, where he took that mixture of chemicals that were in the environment, what we call the primordial soup, and he added heat. that produced a vapor that contained water, but all of the other chemicals as well. Those vaporized chemicals were exposed to a spark that was meant to simulate lightning. The chemical bonds within those molecules rearranged, and then the gas mixture was cooled by adding a condenser. The cooled gas then reformed liquid, and this liquid was sampled with its composition analyzed. Among the components of this heated and sparked primordial soup were amino acids. So again, this does not prove the theory of chemical evolution, but it proves that the chemicals on Earth 4 billion years ago could have produced the biological molecules of which we are comprised. Remember, we spoke about biologic molecules way at the beginning of the semester, and there are 4 classes of biologic molecules: sugars, fatty acids, the amino acids that Stanley Miller's experiment produced, and nucleotides. I've already given the punchline, but it is hypothesized that one of these nucleotides, specifically RNA, was the first biologic molecule. This is inconsistent with Stanley Miller's results, but nonetheless, this is the hypothesis. Remember that RNA is a single-stranded nucleic acid, a nucleic acid that is a chain where sugars and phosphates comprise the backbone, and bases sort of hang out in the breeze to chemically react with other things. It's the single-stranded nature of RNA that lends itself to serving as our potential first biologic molecule. The single-stranded nature of RNA allows it to fold in multiple 3-dimensional shapes. Some of the 3-dimensional shapes that have been identified in single-stranded RNAs are shown here. As I said, the bases that are sort of hanging out are able to form hydrogen bonds with one another such that occasional double-stranded regions can form to create these 3-dimensional shapes. If a molecule can form a 3-dimensional shape, it's possible for that 3-dimensional shape to change. Much like proteins are single-stranded chains of amino acids, single-stranded chains of nucleotides can fold in shapes, and when conditions change, the shapes modify so that RNA is allowed to do work. Specifically, the work that RNA can do is it can function as an enzyme that facilitates or speeds up chemical reactions. RNA is also, as a nucleic acid, able to self-replicate. That is one of the primary requirements for our first biologic molecule. Once we had one biologic molecule, we needed to make more copies of it. That's shown in the top of the right-hand image, where RNA is making more and more copies of itself. RNA then was able to serve as an enzyme, so to facilitate those chemical reactions. And one of the chemical reactions was the production of proteins. So RNA was not only able to make more copies of RNA, but was also to facilitate the assembly of amino acids to make proteins. The proteins then that were produced from RNA's enzymatic activity were allowed to convert, shall we say, RNA into DNA, so that in the bottom image on the right, you can see the present day processes that we use, RNA being the first biologic molecule producing proteins, but those proteins then able to convert the RNA to DNA. Now, once we had those biologic molecules, proteins, RNA, and DNA, it was necessary to contain them. There are many ways that we could have contained cells, but one possibility is the foam that's shown in this image. You may not recognize it as such. This could be foam on waves in the ocean. It could be foam in a drink that you might consume. In fact, a lot of foams are comprised of proteins. In fact, the sea foam, or the foam that's on rivers and lakes, actually is produced from proteins that were left behind by dead animals. So it's possible that protein foams were our first enclosures. However, now we know that our enclosures, the molecules that enclose our biologic molecules to make up our cells, are phospholipids, or fats. So now we've gone from those biologic molecules that Stanley Miller's experiment created, and we've assembled them in a container that now is a primordial bacterial cell, with the primordial soup contained, so those chemicals contained within some sort of membrane. Proteins, RNA, and DNA are within that confined space. Those primordial bacterial cells developed into what are called archaebacteria, shown an archaebacterium here. Archaebacterium still exist on this planet, although what separates them from a normal bacterial cell is that archaebacteria typically like to reside or live in less hospitable places. The Great Salt Lake is an example. Thermal vents, volcanic vents, those sorts of areas. The bad news is, and it's not really bad news for us, but the bad news is that as the archaebacteria are metabolizing those toxic components, they are producing oxygen. So in the ancient world, the production of this oxygen was not favorable for the organisms that lived there. The archaebacteria that are producing the oxygen can't actually live in an oxygen-rich environment. So it was necessary for some bacteria to adapt to that oxygen-rich environment, and this produced our first prokaryotic bacterium, an aerobic bacteria, meaning that it was able to metabolize the oxygen that archaebacteria were producing. So now we've spoken about the production of molecules, the production of primordial cells, and how those primordial cells have evolved to utilize oxygen. But those are prokaryotic cells. So how did eukaryotic cells, those cells that contain nuclei, develop? It's thought that about 2.5 billion years ago, we had an ancestral eukaryote that contained maybe some internal membranes, and maybe those internal membranes surrounded the DNA that made up the genome of this cell. Of course, this ancestral eukaryote was unable to metabolize oxygen, but those aerobic bacteria had adapted to the increasing oxygen concentrations. Well, if I am an ancestral eukaryotic cell and oxygen is building up in the environment, oxygen that I'm unable to deal with, certainly I could die, certainly I could adapt myself, or alternatively, I could live symbiotically, meaning that we could live together such that I could use the oxygen utilizing ability of those aerobic bacteria. In fact, that's what's shown here on the left. A ancestral eukaryote engulfed a aerobic bacterium, and rather than digesting that aerobic bacterium, the ancestral eukaryote maintained that oxygen utilizing bacterium in its cytoplasm. Of course, because it was a bacterium, it can replicate. And now down here, we have a eukaryotic cell with many, many mitochondria. The evidence for a bacterial cell or an ancient bacterial cell serving as a mitochondria include the fact that our mitochondria contain their own DNA. That DNA is circular, much like the DNA of a bacterium. And the ribosomes that are in our mitochondria, and in fact our mitochondria do have ribosomes, those ribosomes are of the same size as the ribosomes in a bacterium. So revisiting our evolutionary time scale, we see that Earth developed about 4.5 billion years ago. It took about another billion years in order for life on Earth to develop, but that life on Earth was in the form of a primordial or an ancient bacterium. That bacterium was similar to our archaebacteria, but those archaebacteria produce oxygen that other bacteria had to adapt to. Ultimately, some of those oxygen-utilizing bacteria were engulfed to create eukaryotic cells. Those eukaryotic cells and the bacteria lived together on Earth for quite some time before multicellular organisms developed about 600 million years ago. So to recap our evolutionary tree, we started with our ancestral prokaryote. That ancestral prokaryote developed into an aerobic, an oxygen-utilizing bacterium, as well as anaerobic ancestral eukaryotic cells. So that's here. Those once the oxygen concentrations in the environment grew substantially, eventually these anaerobic eukaryotes had to engulf an oxygen-utilizing prokaryote that became our mitochondria. In fact, that ancestral eukaryote subsequently was able to develop into plants, animals, and fungus. We're going to focus on the development of animals, since this is a human biology class. So we had ancestral eukaryotic cells about 2.5 billion years ago What had to occur in order for us to develop a multicellular organism like ourselves? We're going to talk about the development of animals, the development of animals with backbones, and the development of mammals. Finally, we'll talk about the development of primates and, in fact, what a primate is. All of these things are part of human development or human evolution. So here is our tree of life looking at bacteria on the left-hand side and eukaryotic cells on the right-hand side with our archaebacteria in the middle. That suggests that all of us sort of came from these archaebacteria. This is a multicellular organism that is thought to have been sort of a halfway house, as it were, between single-celled organisms and multicellular organisms. This organism lives part of its life as a single cell, and when conditions are, I'll say, appropriate, those single cells collect to form a multicellular organism that generates spores. A second example of a sort of a halfway between single cell and multicellular organism is this colonial organism that's called a volvox. This lives in pond water, and it's comprised of a number of different cells that each do their own thing. However, the cells live together, sort of as a colony. And again, when the conditions are right, some of the cells in this colony become differentiate, so that they can create sex cells, so that these volvox colonies can reproduce. We'll focus on the development of animals from these sort of halfway in between organisms. This is an evolutionary tree of animal evolutions, starting with a single-celled animal ancestor. And as we move up the tree, we see that a number of things had to occur in order to produce the animals that we're familiar with. The first step, of course, was to develop multicellularity. So we needed to develop multicellular organisms. Following the development of multicellular organisms, like the ones that I showed you on the previous slide, we developed some ability to differentiate such that we had multiple cell layers, starting with two, the ectoderm and the endoderm, and ultimately landing on three tissue layers, those that we spoke about during our developmental biology unit. So we have three tissue layers: ectoderm, endoderm, and mesoderm. And all organisms that came from these three layers are within the parentheses. So flatworms, roundworms, and all sorts of other organisms, including chordates. Chordates are animals with a backbone. Here is an evolutionary tree, again becoming more complex as we move up the tree, of animals with backbones. We start with the jawless fishes, so those are the first ones that have a backbone. And from those jawless fishes, we develop a number of other animals that contain backbones. We are among the amniotes, animals with backbones, whose young develop within an amniotic sac. Now, in some cases, the amniotic sac is surrounded by a hard shell, so an egg, much like those of reptiles and birds. This is the evolutionary tree of those amniotic mammals. Squared here are the primates from which we have evolved. Primates, this is an evolutionary time frame of primates. And primates, again, this is a class of animals in which humans are located. Primates have a number of traits that define them as primates. The first of those is that primates have a large brain relative to their body size. They actually have a fissure or a crevice in the middle of their brain. That's called the central sulcus, and that is shown in this diagram. So all primates have a brain with this central sulcus or fissure. Primates also have specialized cells in their skin for tactile or touch sensation. These cells are called Meissner's corpuscles, and they allow us to sense things very astutely through our skin. And finally, primates have what is called a decussating optic nerve. What that means is that the vision that's coming in on one side of our head, so let's say into our left eye, crosses over and communicates with the right side of our brain. Conversely, information coming in from our right eye crosses over to the left side of our brain. This crossing over from one side to the other is what decussating means. Finally, I'll conclude with evolutionary evidence that eggs came before chickens. So which came first, the chicken or the eggs? On our evolutionary timescale, eggs existed well before chickens. This is the conclusion of our sort of human evolution discussion. I want to talk next about some of the underlying premises, the things that drive evolution, and finish that with molecular evolution and the contribution that molecular evolution has on some present day processes like the development of cancer.